Good evening and welcome to our Prader-Willi Research Foundation Presents webinar, where we're aiming to connect you with world-leading research on Prader-Willi syndrome in a way that's accessible and practical. I'm Kathleen Jones and I'm the founder and CEO of the Prader-Willi Research Foundation Australia. For those of you who don't know our organisation, our work focuses on growing research into treatments for Prader-Willi syndrome and into growing access for Australians with Prader-Willi syndrome to those treatments. This evening, I'm really delighted to welcome Professor Giles from the University of Cambridge, a neuroendocrinologist and a world-leading expert on obesity. I'm also delighted to welcome Associate Professor Tania Markovic, endocrinologist and Director of Metabolism and Obesity Services at the Royal Prince Alfred Hospital here in Australia, in Sydney. And Dr. Markovic leads the Prader-Willi Clinic. Well, thank you both for taking time this evening to discuss obesity and to discuss Prader-Willi syndrome.
A pleasure.
Yes.
It's a real treat. Thank you. And look, it's great to welcome our live audience. Remember that the live chat is open, so you can ask questions on the chat, and I'll do my best to put as many possible of your questions to our guest tonight. So let's move. Let's kick off. Professor Yo, you've been spearheading some really groundbreaking research on obesity. How did you start working in the area of obesity?
I started working on the area of obesity by accident, as these things do. I was trained in genetics. I did my undergraduate in genetics in California, which is where I'm from, and then I went to Cambridge to do my PhD in genetics and molecular genetics. And then it's one of these things where I finished, and I ended up in the lab of Steve Ratley, just by chance, working on the genetics of severe childhood obesity. And in this particular case, we were looking at cases of childhood obesity where single gene mutations cause really quite severe hyperphagia and hence severe obesity. And we worked through this would have been 1998 or so that I did this, and we identified a number of genes within the fat sensing pathway of the brain that when mutated, single genes that when mutated cause really quite severe hyperphagia in kids. And so I started working on that and have been in the field ever since professionally.
That's great. And when did it go from being a job to being really a passion for you?
Yeah, that's so. Yeah, you always start these things as jobs, particularly in your early 20s. And what I'm in Cambridge. I don't know how many of the audience here have ever visited Cambridge. But I think actually, if you've ever watched any Harry Potter, it really quite does support. It's sort of like you got these long benches. I have to wear black robes, that kind of thing. The candles are not floating. That's the only thing that's not real. But I remember this actually. To this day, it must have been 2000, 2001. I was still quite young as a postdoc, and a senior colleague of mine brought me to some fancy dinner, and so long benches, and I was sat at this fancy dinner, and sat opposite me was this guy. He was a beardy older white guy who was a professor of something or other. And as you do at these things, he went, "What do you do? What do you do?" And I told him, I said, "I study the genetics of severe obesity. I study the genetics of severe hyperphagia." Is what I said. And he went like this. Okay, I'm gonna exactly the motions. He says, "Do you know what your problem is?" Like jabbed his finger in my face. I said, "No, I'm trying to have dinner." And he went, "You give," and he said it exactly like this: "You give fat people an excuse." I was going, "Oh, dude." So dinner was ruined, but then I tried to sort of explain to him, I don't know how convinced he was. I was in the cab on the way home, and I was thinking to myself, listen, this is a professor from Cambridge with alphabets after his name and a supposedly well-educated person, and this was his opinion of obesity. Okay, and so it was at that point I realized that, you know what, I can't just work on it on a day-to-day in the lab, because if I do this in the lab, but I don't confront what people I would say was at the time very, very prevailing view, and now still quite prevailing view in society, that whenever you study obesity, whenever you're trying to study the genetics of it in particular, suddenly you're giving someone an excuse. And I realized then that I needed to take any opportunity beyond speaking to the bubble, to my academic bubble, my other colleagues, to make sure that I do events like this. Okay, that I go out and actually speak to people either with lived experience, which is obviously hugely important, or more importantly, the people without the lived experience, in order to spread the message that, listen, there is a lot of biology in play when we're talking about body weight.
Thank you. Thank you. There is Dr. Markovic, I wonder whether this story resonates with you at all. How did you become specialised in the area of obesity?
Look, unfortunately, the story doesn't resonate. It's not what happened to me. I was working at the Garvan Institute and at St Vincent's Hospital. And when I started there, they said, we're not interested in type 1 diabetes, we're interested in type 2. And then when it came to doing my PhD, it was suggested that I look at diet and type 2 diabetes and weight loss. And that was the basis of my PhD. And I guess then I saw the dramatic effects of both eating less calories and losing weight on diabetes. Not a lot was known back then. That was the 90s. It's interesting. In fact, some of what I did then, people are discovering now. They think it's new, but anyway, that's another story. And then I guess from there, I was offered some work at Royal Prince Alfred, where first at Sydney Uni, then Royal Prince Alfred, where the first obesity service was set up by Professor Ian Caterson. And he mentored me, and then I ended up working there as what's called visiting medical officer. And then and through my work, I mean, it's very apparent how important weight is in contributing to a lot of diseases. And what struck me in Prader-Willi syndrome is how, because weight loss in people with long-standing diabetes isn't always, you don't always reverse diabetes, but I know I want to do some more than anecdotal research. I actually want to look at our clinic's data, but to me, what I see is that people with Prader-Willi who can lose weight, a large amount of weight, their diabetes goes away, even if they've had kidney problems, with albumin leaking from their urine, which is from their kidneys, into their urine. This all disappears when they lose weight. They seem very responsive to weight loss for conditions like diabetes but also for their breathing problems, etc. More responsive, maybe because they're younger and so their bodies can recover better than the older people I tend to see. But it just demonstrates how important it is to reduce weight to control disease.
Thank you. Professor Yeo, we all know what it is to overindulge in food. So can you help us to understand obesity and what's known about the cause of obesity in general?
So the cause of obesity, okay, there's two answers to this. There is the simple answer, and actually is the answer that people mostly will say, and that's that obesity is because people eat too much. And from eat more than they burn anyway. Now, from a physics perspective, that is undoubtedly true, and that is how you get to the body weight has to be a function of physics because it's a fundamental law. But that's not where the fulcrum is. That's not where the interesting biology lies. The interesting biology doesn't lie in the how; it lies in the why. So why? And I'm a food intake person. So why do people behave so very differently around food? So, for example, why do some people respond to stress by eating, whereas other people respond to stress by not eating? Okay, you can divide the world into two. Same hormone goes up, cortisol, but yet some people respond to it by eating and other people stop eating. Okay, why? Okay, there's going to be some genes there. Why do some people love food? I'm a food lover, whereas other people use food as fuel. Okay, why do some people appear to take more food to get full up? Why do some other people, appear to get hungry more frequently? And these are not imagined behaviors. They're just not imagined behaviors. Now, clearly, why, where, what, and how much we eat are going to be the function of, huge cultural and sociological underpinnings. How can it not be our food, right? But there is huge biological underpinnings. And now we know and understand that the genetics of body weight on one end of the spectrum just happens to sit obesity, is the genetics of how our brain influences our feeding behavior. So to put simply, why there is a percentage of people who end up with obesity is because they're more driven towards food ultimately, and because they're more, for a myriad of different reasons, not just because they're more hungry. There's going to be a whole host of reasons. In fact, I got this analogy which I use, okay, which is the Monday morning meeting scenario. Okay, now you go on everywhere. We've been these things. We go on Monday morning. We have to sit around a table. Okay, and suddenly someone has brought cookies to the meeting. All right, and I would argue that there are at least 4 different behaviors in response to the plate of cookies sliding down the table. First response, okay, and this is me. Someone's already picked up the cookie and started eating it before the plate of cookies has stopped sliding. Okay, that's me. Okay, so you eat the cookies. Oh my God, delicious. Then you have a second type of people, the people who are long for the cookie. They want the cookie, but for whatever internal algorithm, they don't pick up the cookie. Okay, behavior number 2. Behavior number 3. Okay, now these are the people who don't even remember eating the cookie, but eat the cookie. How's the cookie? Am I eating a cookie? Right? And 4th, they're the people who don't care about the cookie at all. They can come by and they're going, what cookie? They're already listening to the meeting. Now, there are more behaviors, but that is what feeding behavior is. You're going to have people who are obsessed with food. You have people who eat absent-mindedly. Suddenly you sit on the seat watching TV, and oh my God, I finished 3 packs of biscuits and a thingy of popcorn, right? Then you have the people who so obsessed with food, but because of internal control, won't eat the food, but then they can't think of anything else but the food. Okay, these are the people with the food noise. I'm going there, etc., etc. They're genes that underlie all of these behaviors. There's environment that underlies the behaviors as well, clearly, but they're powerful genetic underpinnings. So that is the issue, right? You get people and they're not mutually exclusive, and we all know people who behave in these fashions. And that's what I study. I study how the brain influences these feeding behaviors beyond simply just hunger, to be true, which also exists, obviously.
Look, our families that live with Prader-Willi often describe the hunger associated with Prader-Willi as painful rather than really seeking the pleasure of eating. How does that appetite mechanism work?
So, okay. So look, I think appetite is an interesting word, right? Because you think, well, what is the concept of appetite? And appetite is an interesting word because it's an integrated term. So it in of itself, it encompasses really quite a complex series of behaviors, 3 in particular. Appetite encompasses hunger, it encompasses fullness, which is different from hunger, different circuits, and it encompasses the joy of eating. All right, that is over there. What makes it taste nice? If you imagine a triangle in which appetite is actually set in the middle, you can imagine that if you tug on one of them, if you're really, really, really hungry, then the simplest foods are delicious. A piece of bread, a bit of cheese, a bit of rice, right? If you're very, very, very full, then you're not hungry at all. And then you need really high energy dense foods, rich in fat, rich in sugar for you to want to eat it. This is the physical manifestation of your dessert stomach, okay, when you're there. And so that is what appetite is. Now, what happens, however, is if you have a severe genetic tug, either you pull the hunger so much that everything else goes awry. And so we've seen genetic causes, I mean, in and amongst, including Prader-Willi syndrome. Like, for example, there are these kids with something called leptin deficiency. So leptin circulates in proportion to the amount of fat, okay? And it signals to the brain to let your brain know how much fat you have. There are very, 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 very rare cases. There may be only 80 cases in the world, super rare, okay, of kids with mutations in leptin. So in other words, they have a brain who can't sense leptin, and so hence think that there is no fat south of the neck, okay? And so when don't you have any fat? You don't have any fat when you're starving. And so these kids eat like they're starving. There's hyperphagia. These kids have to have their freezer doors locked up because the kids will go and actually eat the frozen food. They'll eat. There's no really ability to discriminate between high energy dense foods like a pudding or like a cake versus broccoli. Okay, they'll eat just anything. because they have a brain that thinks that they're starving. And so what happens there is you're then in a situation when you're truly starving. You don't eat for the enjoyment of eating. You eat because you think you're starving, and so therefore you're trying to actually push down the starvation. In the situation of leptin deficiency, the problem is they can never get that. They can never get that feeling of joy and fullness because they will always think they're starving because their brain can't sense the fat. Ultimately, that is very, very rare, but we know of mutations throughout that entire pathway. So that is hyperphagia. And when we're talking about what I study and within Prader-Willi disease, that's a different disease, but it influences part of the same part of the brain, the hypothalamus. Okay, and so if you have, we don't actually know what Prader-Willi does to the hypothalamus is the answer. Okay, but you can imagine there's something happening there that probably prevents it from sensing certain cues that are coming up, and hence it mimics what we see in leptin deficiency. Your brain thinks you're starving, so you end up eating without the joy because you can't help yourself because you think you're starving, and so therefore you have to eat.
Well, look, can you tell us a little bit more then exactly about the work that you are doing to understand the hypothalamus and to try and help us understand what is happening in Prader-Willi syndrome?
So one of the key questions I think to ask within Prader-Willi research is there common ground, common soil, shall we say, between both the severe obesity and the severe hyperphagia, and other causes of severe obesity and severe hyperphagia? And you might think, well, why do we care? Why? Well, we care because if there are going to be Prader-Willi specific pathways that are changed, okay, now this can be genetic or this can be structural, then we have a chance to say, okay, well, this is a Prader-Willi specific change. Can we design something to actually target that specific network? So that's one thing. But if there is something shared, if there is a shared soil between the two, then you can ask the question, well, is the non-Prader-Willi obesity or whatever that is overlapping, is there already a drug for it? Is there a system that can actually tack that? And so we're doing it in two ways. We are looking for novel genetic causes of obesity, and we've just found one actually. It doesn't really matter the name of the gene, but it's called SREK1. And what is interesting about that gene is we found it in a that when mutated, and we did some fancy schmancy stuff by putting in the stem cells and converting into neurons and looking at the stuff, it downregulates, okay, globally, really these genes called SNOD RNAs. I don't know if you know what these are, including SNOD 116s, which we identified that when you actually have a mutation can cause Prader-Willi syndrome. And this mutation, we had no idea that it had anything at all to do with Prader-Willi syndrome, suddenly downregulates all these SNOD RNAs. And then when we phenotyped these individuals with SREK1 mutations, then they don't have Prader-Willi syndrome, but they have elements of Prader-Willi syndrome. They have the initial hypotonia, and then what that happens is after you get the catch-up weight gain, they then ended up with hyperphagia. They do have a little bit of cognitive defects and cognitive delay, difficult to phenotype. There's a skin picking. There is a number of different things that are there, which is not Prader-Willi syndrome, but with common soil. And so we're now trying to actually figure that out. So that's one element. On the other side, what we're trying to do, and I don't want to sound morbid, but it has just been accepted. We have been trying to map the feeding circuits within the hypothalamus of humans. Okay, so donor samples. And at the moment, we have constructed a 3D structure of the hypothalamus with all the feeding circuits, including where Ozempic binds and things like that, in the normal weight hypothalamus. All right. But now we're working on the hypothalami from severe individuals, and we're working on the hypothalami from donors who were diagnosed with Prader-Willi syndrome. And the goal there is to look at the structure of the normal weight hypothalamus, look at the structure of the obese donor hypothalamus, and then look at the structure of a hypothalami from someone with Prader-Willi syndrome and compare them. Look at what's common, look at what's different, look at the genetics underlying it, and see if we can identify common soil or unique drivers for Prader-Willi syndrome.
Good. That's so interesting. Do you thank you. Look, I just think before I ask any more questions, we should also just acknowledge that the Foundation for Prader-Willi Research has at least partially funded some of your work on Prader-Willi syndrome, and we're very grateful for them to have done that, obviously, because we're all beneficiaries of this wonderful work. So do you think then, looking forward, what is the likelihood that you think we'll actually be able to find treatments?
Oh, no, I think the likelihood is high. Now, the issue is, however, until the main problem and why there hasn't been a treatment yet, I'll be frank with you, is because there are no good mouse models of Prader-Willi syndrome. There are mouse models of Prader-Willi syndrome there are mouse models which replicate elements of Prader-Willi syndrome, bits of it, but not all the phenotypes, because mice are mice and humans are humans. A lot of the other conditions that I work with, we have good mouse models, which means that you can try drugs, you can do all kinds of things to it. You can accelerate the research. Now, look, no one's going to want anyone to inject anything into your kids with Prader-Willi syndrome, understandably. So it's been delayed because now you've got to use cellular models. You have to use a number of different adjacent methods in order to actually identify. But listen, the moment we understand more about what we know the genetic cause of Prader-Willi syndrome, we don't know the mechanism underlying Prader-Willi syndrome. The moment we realize what the mechanism is, where are the key nodes that are an issue, then we have a fighting chance of actually either designing de novo drugs, so drugs that purely for Prader-Willi syndrome, or more likely repurpose drugs that are already out there for treating obesity, specific types of drugs, to actually target Prader-Willi syndrome. Now, I am very, now that we have the technology, AI and all these things, and people said, well, why are you saying AI? Because it's true, because then you can sort of do multiple iterations of these things. I do think that the opportunity to find a treatment, not for all the Prader-Willi syndrome, but to focus on I focus on hyperphagia. Do I think there will be a drug that can at least mitigate a significant percentage of the hyperphagia seen in Prader-Willi syndrome? Yes, I do.
Well, no, there's probably every family listening to that will have a smile on their face. It's very heartening. I wanted to ask Dr. Markovic now. Your work, Professor Yeo talked about Ozempic and how it lightens up some of the brain, or the hypothalamus, some of the pathways. Can you tell us a bit about your work and what you're doing with tirzepatide?
Yes, so we received some money from the Prader-Willi Foundation, which is we're very grateful, of Australia, to look at tirzepatide, which is similar to Ozempic, which is semaglutide, in that it also has this glucagon-like peptide 1, and it's got a second gut hormone, GIP. It is on the market in Australia, in the US and Europe for the treatment of type 2 diabetes and obesity, and is the best drug available, or results in the most weight loss of any drug that we have, where the maximum dose on average in people with obesity who don't have diabetes. People who have diabetes often lose less weight, which is interesting in itself, but people who have obesity without diabetes are losing on average 22% of their body weight, which is amazing, and having health improvements, importantly. So clearly this is something that we'd like to see if it helps people with Prader-Willi syndrome. But we also want to make sure that it's safe. So we're doing a short study, just a 3-month study. And this drug, as a lot of the sensing, as Professor Yeo was saying, sense nutrients in the bowel. These are gut hormones that can affect the bowel. They can result in delaying gastric emptying. They affect motility of food further going through the bowel. And these are pre-existing problems in a lot of people with Prader-Willi syndrome. So we most importantly want to look at whether they're safe. I don't know if Professor Giles probably knows this, but if people with Prader-Willi, if they have a huge binge, they can sometimes suffer this terrible condition where the stomach can rupture because of their very slow gastric emptying, which is, can be devastating. So the first thing we want to do is make sure that these medications are safe. And then at the same time, in 3 months, we should be able to see, because we're going to escalate the dose quite quickly to the maximum by the 3rd month. And incidentally, when we've used similar drugs in people with Prader-Willi, they seem to be very well tolerated. So my hunch is that we won't see any major safety problems. And I'll be very curious to see if it helps. So we're we're pushing through ethics, because we're using a capsule that's been developed in Australia called an ATMO capsule that we're measuring gastric emptying with the gold standard what's called scintigraphy scanning with a radiopharmaceutical, but we're going to be looking at about small intestinal and large bowel function with this capsule that measures the carbon dioxide and oxygen in the bowel, and that tells us how it's moving through the bowel. So we have to, anyway, we're working through ethics. Want to make sure it's safe for our patients, of course. So I'm very excited. I hope to start it next year. I was hoping to start it this year, but things move slowly. So early next year is the plan.
Look, it's great for us. I can't tell you as a parent how important it is to hear that we've got, potential treatments coming through that can help our kids. I just wanted to check in with the audience online whether there are any questions before we keep moving on.
While they're thinking, I'm going to ask a really basic question. I'm so embarrassed. Don't tell anybody, Giles, that I'm asking you this question.
Okay.
So you're doing this very interesting work looking at the circuitry, which to me makes so much sense. And your group has discovered things like the melanocortin 4 receptor abnormality, which is very common at what did you say yesterday? 0.3%.
Yeah.
And even a higher, so I think it's something like, correct me if I'm wrong, 3 to 5% of people with early onset obesity have an abnormality of this receptor that is very important in appetite control. How did they actually find that the receptor was defective? They didn't look at brains to work that out?
Oh no. So we didn't do that. We looked at genetically, which is obviously, then it means that you can do it from a blood test. And we stopped.
So is that melanocortin 4 receptor in the blood?
No, we looked at the gene.
In the brain.
We look. It is in the brain. It's specifically in the brain, but we looked at the genes. We screened the genes to find the mutation, and then we cloned the receptor, and looked at it recombinantly to actually find out that it is pathogenic, so that it doesn't actually work. So you do a genetic screen, which means that you can just do it from a blood or spit, and then what then happens is you then find mutations in these genes, and then you put them into a cell, and then you sort of functionally test to see whether or not they're dysfunctional, and that's how we find out whether or not they're dysfunctional.
Right, and because we know that the only gene that's is there any point? Can you do something like that in Prader-Willi?
The problem with Prader-Willi syndrome, so this is what we're trying to do, actually. So the problem with Prader-Willi syndrome is, we know it's an imprinted. I don't want to teach you guys what's happening. We know it's an imprinted condition. The major issue is because there's so many genes in that imprinted region. It's not just one gene. And the issue is, which of it is it? I mean, my pet hypothesis is actually the SNORD116s are probably actually a key driver, but we don't know what the hell they're doing. What are they binding? Are they changing the expression of a whole host of other genes, for example? And so we've been using gene editing in order to sort of edit increasingly large deletions of just the SNORD116 region into stem cells, for example, because you can't just take those genes and put them into a cell, because the MC4 receptor is very small, so you can clone it, you can muck about with it, and it's quite easy to move. It's very portable. When you're dealing with Prader-Willi syndrome, the region is way too big, it's way too complicated, and so you have to use the endogenous gene, so the gene that's actually present within. So we have human stem cells, which we can differentiate into hypothalamic neurons. We can differentiate into anything, actually, but we can change them into human neurons in a dish. You can then edit in mutations in Prader-Willi genes. I focus on SNORD116, for example, and then you can ask, well, what happens when I actually mess that up? Can I actually see changes in expression of other genes that can give me a clue as to what the SNORD116s might be doing?
Very interesting.
And at some stage, will that, just thinking, Professor Yeo, about your comment on mice models, at some stage, how do we solve understanding the phenotype or the phenotypic expression of SNORD116? Is that coming back to having to develop better mouse models?
So, I mean, I did do it in 2018. I actually published a paper in which we knocked down. So it's difficult to remove SNORD116. Well, if you remove SNORD116s from the whole mouse, which you can do genetically, you get quite a sick mouse. Actually, nothing. The mice are small. Not a great big deal happens to it. So what we ended up doing was we ended up removing the SNORD116s from the adult mouse, which requires injection into the mouse to knock down the gene and put it up. And there we replicated the hyperphagia in the mouse. And so and that's actually one of the big reasons why I ended up. We identified that SNORD116 deletions. We and collaborators, not me personally, not me solely, identified that SNORD116 deletions can actually lead to a huge overlap with Prader-Willi syndrome and actually Prader-Willi syndrome. And then that was the next thing we ended up doing. We knocked it down in mice, which says, look, this looks like it could really be the case. Then I hadn't touched it for a while until we found this new mutation in this other gene, which suddenly showed also these downregulation of SNORD116s. So I think we're gonna have a number of different ways of trying to get at the problem, I think.
And so say you do identify that SNORD116 is a key gene for hyperphagia, how do you then go about identifying the drug?
So I don't think that SNORD116 in of itself is going to necessarily be the target. I think we're going to need to find out what SNORD116 targets in of itself, because the interesting thing about SNORD116 is it's a gene, but it's not a protein. So you obviously have genes that become proteins, and the protein does something, all right? Whereas SNORD116 always stays as an RNA, and we've now had gone through COVID, and we know that you can convert that, and it stays as an RNA and functions as an RNA. Okay, so what is it doing? And so the likelihood is, because it functions as an RNA, that it will be changing the way other genes are turned on and turned off. So we're trying to identify which of these genes are turned on and turned off, because I think this is a hypothesis, but I think it's going to be 1, 2, or 3 of these genes that have their on and off switch screwed up, that are going to hold the secret to where we might find a potential anchor, a potential handle in which we can go in and actually do something and try it anyway.
Wow, that's look, I can't wait till you find that gene. And then, I guess for us, then once we found it, what do we do? Once you found the targets of 116, then it comes back to drug repurposing or de novo drug development.
Exactly. Now, then it entirely depends on what we've found. Okay. So I think it's going to be more complex than 1 or 2 genes. I bet you it's going to be a whole stack of genes. And then we're going to have to look through that whole stack of genes and figure out, well, are any of these bona fide targets? Okay. Or are any of them already implicated in other causes of obesity that we have gone through? And so that is the way. Look, I don't want to, I'm managing expectations here. I think we're moving one step at a time in order to try and do it. I am highly confident we will find something. I don't know when. It's not going to be next year, but I am confident that we will find something that gives some hope.
That's great. Thank you. And look, in the meantime, whilst we're looking for, for drug development, what are there other things that families can be doing, given that if we take this brain lens, do things like neuroplasticity offer us any options? We see that there are projects working on mindfulness. What are some of the options that we might have, or is it really a matter of waiting for those medications?
I think I'm going to answer from the perspective of non-Prader-Willi obesity first. And the answer for non-Prader-Willi obesity is it depends. And largely, simplistically put, the more severe the obesity, the less likely that a behavioral change will solve significant amounts of the problem. This is my view. Okay, so if you consider that at the very, 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 very severe end of non-Prader-Willi obesity, I do think that surgery is still going to have to be the answer. Non-Prader-Willi obesity. I think you're going to get to the situation like myself. My wife thinks I should lose a few pounds. I probably do need. No one's going to give me Ozempic. I need to have behavioral changes, right? So for me, some kind of mindfulness-y, thing that we're going to go, whatever would work for me is going to be the appropriate thing. However, we now have drugs that will probably work for a large number of individuals that sit in between people who are trying to lose 5 kilos and someone who is 150 kilos overweight, okay, in severe obesity. And there probably sits the region where some mix of behavioral modification together with some of the drugs that are available. That is non-Prader-Willi obesity. Now we come to Prader-Willi obesity, and you're also going to get a similar variation in severity of different aspects of it. Okay, I'll focus on the hyperphagia because that is my area of expertise. There are going to be some people with Prader-Willi, some of you with kids with Prader-Willi syndrome who are going to be very, very severely hyperphagic, and there are others who have their hyperphagia more easily actually controlled. Okay, we actually don't know why this is the case. It's not necessarily the size of the deletion. That there's going to be some other reasons. So there's going to be two things. Can we understand this phenotypic variability, what we call phenotypic variability, number one? But number two, then we do have to sort of treat the person in front of you have to treat the person in front of you, which means that, look, if Joe over here has really high severe hyperphagia, then we're going to have to do something beyond just behavioral modification. Whereas I think if the hyperphagia is not that strong, then I think behavioral modification will always have a role to play, in trying to actually tackle the problem. Is that an answer? I don't know if that was a fluffy answer.
That's great. That's great. Tania, I'm wondering if you've got anything to add to that.
Look, not really. I mean, the only thing that I would add is that in 2024, the behaviour that we've got to modify. Well, we've got to modify the environment, and you referred to it, Giles, earlier when you were talking about the children with leptin deficiency, and so we do need to make the environment. We do need to. Ideally have a kitchen we can lock or a fridge we can lock or something, so that which I think a lot of people with Prader-Willi feel much less anxious if they know that they're aware that they have to control what they eat, and I think they feel, even though it sounds very restrictive and an impingement of your liberties, but they actually feel a lot better knowing that the food is away, and if they have set patterns of when they can eat and what they can eat, when there is food there that shouldn't be there, that upsets them, because they know that, a lot of the patients it upsets, not everybody, because they feel stressed because they really want to eat that food and they know that they shouldn't. So that sort of modification of the environment in 2024, I think, is still very important, and it's our best, it's sadly still the best treatment that we have. I look after adults. I see a range of adolescents who are coming to our clinic, and there's quite a variety. I mean, some of the parents have just been amazing in how they've managed these children, and the kids, it's phenomenal. And then others where there's been a bit more dysfunction in the family, or, there's been things happening, and maybe parents haven't fully understood, or the diagnosis has been made very late, and then it's much harder. The child's missed out on growth hormone. That's really changed the trajectory of people with Prader-Willi syndrome. That's very interesting, other aspect. So you see quite a range when they come into the clinic, and it'll be interesting to see how these young people who have had the benefits of growth hormone, whose parents have been very well educated, whose parents have managed to do, despite all the stresses that go on in one's life, managed to really help their children, how they will progress. Whether it'll be different to a lot of the adults I see, who really the only way that they end up having reducing their weight and having better health is by living in a Prader-Willi specific home where there are carers who ensure that and again, it varies if the group home is very well educated and you don't get a change in staff and the patients have a good programs or they're working, their lives will be a lot better. So currently having the right environment, the right parents seems to be what's most important. It's going to change. It's going to change when your work comes through, Giles, and maybe my study.
Look, we do, some doctors and some families that we've spoken to observe or have made the observation that food seeking itself over the lifetime, so we're thinking 20 years, is less in people with Prader-Willi who've had minimal sweet flavors throughout their lives. It's quite fascinating for sort of me as a parent to think about that as a potential intervention. I'm just wondering if you've seen anything that might, currently this is anecdotal. So have you seen anything that might actually shed some light on this?
Me or Tania?
You, Professor Yeo.
So just to be clear, I'm a molecular biologist, so I don't tend to study whole human beings. So I don't know the answer to the question. However, I will proffer an opinion because I'm an opinionated individual by way of an anecdote, if I might. So when I was in my early 20s, I've got colleagues who study hyperphagia. I've been in this field for 30 years. And the way they test hyperphagia, so specific types of hyperphagia rather than the Prader-Willi, so specific genes, is they set up sort of a buffet meal scenario for kids. It's a controlled situation, so they can't hurt themselves. They come in, everything is measured in, everything is measured out, okay? And then they're trying to see how much you would eat. They're trying to measure the scale of the hyperphagia. But in order to do that, you need to put enough food on display for the dynamic range. And so myself and another early 20-year-old, so people who can eat a lot, okay, was sent in to test out this meal. And it was an enormous meal. But what was interesting, so our instruction, which is kind of instruction that you never get, is to eat till you're full. I said, ooh, okay, or eat till you can't. I said, ooh, okay. But what was interesting was when I went in, all the food was bland. Okay, there was no ketchup, tomato sauce. There was no mustard. There was no condiments. There was no, we were given water. There was given this. And the reason behind that, because then I came out and I ate. So I could eat, and I ate quite a bit, and I walked out. But if I had barbecue sauce, if I had some condimenty type thing, I would have eaten a lot more, right? Whereas because the food was bland, I didn't. So I'm going to convert that anecdote, and this is to my mind, and this is why the test was set up in that sense. They were trying to measure just baseline hyperphagia eating without the enhancement of everything that's there. The problem, obviously, is all the food now is enhanced. So the issue is this what this anecdote is pointing out, that the sweetness of it acts as a flavor enhancer. And because it acts as a flavor enhancer, remember my triangle, which I'm telling you about, where you get fullness and hunger and reward. And suddenly you throw in sugar, whatever other flavor enhancer you have, and it really lights your brain up like a Christmas tree. Then you end up eating more, and you learn that, oh, wait a minute, food means that I'm going to get that more. And you start to poke it more. Maybe that's the case. I don't know is the answer, but that would be if I had to guess, that would be the way that I would guess it worked.
Look, I find that so interesting because I do, personally, my anecdote is that I see a child who will have a much heightened response to food if she knows that there's some kind of sweetness involved, and if she,
don't we all
sweet things drive that.
Don't we all? And this is the thing, right? We don't want to say it like it's some weird Prader-Willi thing, because don't we all? I mean,
we don't all. Some people like sweet things. Some people like salty things.
Some people like fatty things. You're absolutely right.
I don't like sweet things. I will have cheese. I will not stop eating cheese. And my mother was the same. So I think it's genetic. But I think what you're exposed to, because I know that as a child I was able to drink Fanta, and which is an orange drink that was incredibly sweet. I think they make it a little bit less sweet now, and lemonade. And still like my cheese back then. This is very anecdotal, but I think there is some evidence to support it. And I haven't had those sorts of things for ages. And now when I occasionally, once I picked up a drink and I thought it was mineral water, but it was lemonade, and I just couldn't drink it because I'm not used to having that sort of sweet stuff that I had. And I think maybe it is that stimulation of the the hedonic part of our brain or whatever. But I think you do get used to sweet things, and then the more sweet things you get, the more you get used to it. And whether this happens in Prader-Willi, I don't know. But I think there is, again, it's not my area of research, but I think I've heard it at research meetings that there is something to support that.
Can I add something else? Actually, I went to a meeting. This is something which just jumped into my head, as these things do. I went to a meeting called SSIB, which is an American-based meeting, the Society for the Study of Ingestive Behavior. Now, I was invited to speak, and so I was speaking about something else. And I was sat in the audience, and there was someone actually measuring sweet, the likeness for actually sweet things, and particularly in baby mammals. And what this particular talk I went to showed is that actually baby mammals of all flavors, not just human babies, love sweet things, ridiculously sweet things. Okay, I mean you've tasted. I mean, I've tasted some of the food that children eat and go, oh my God, my pancreas is dissolved. What is that, right? And what then happens is, this is something that's hardwired in all of us, because what is the role of a baby mammal? Well, the baby mammal, once it pops out, is to make sure you latch on to the highest density source of fuel when it comes out, which is the boob. All right. And then you actually have fat and sugar. Lactose is a sugar. Fat and sugar. Why? Because you want to avoid becoming tiger food. Then what then happens is, at some point, as you hit puberty, typically, your palate matures. Like, for example, I hated coffee prior to 12. Now I drink black coffee. Like, what the hell is that? Or beer, right? With the first time you drink beer, how does anyone drink this? But then your palate matures. So is there something to be said for a delayed maturity of that moving away from sugar, or does having the sugar delay the maturity into something that is that slightly differs? Once again, I'm just telling you from a perspective of evolution, of the hardwiring. Incident to anything else. This is why we need to protect kids, all kids, from sugar, because they're hardwired to eat the sweetest things available if you kind of leave them alone. And so we have to protect them from themselves.
Interesting.
Look, you're a man after my own heart, Professor Yeo, because we often my board and I talk about what is the role of Prader-Willi in understanding obesity more generally, and what can we contribute as a research organisation, not just to advancing treatments for Prader-Willi syndrome, but to making a change for obesity in general. And clearly, childhood obesity is a big issue that needs to be addressed.
It is a thing that we need to address. It's rising all over the place, and we need to tackle it, because I think, look, if anything, because unhealthy children become unhealthy adults. I mean, primarily that's going to be the big reason. So we do need to do something about it.
I think we've just had a question come in, if you don't mind me asking. It says, in addition to genetics, do you think that microbiota has a role on obesity in Prader-Willi syndrome?
I think the microbiota is, for people, this is an interesting question. People ask me, is the microbiome research, is it good science? Is it bad science? I think it's new science. I think is the answer. So what do we know? I'm just answering in general first. What do we know about the microbiome? I think we know that you need it to be healthy, and it is a wonderful reflection of your environment and of your diet. Now, which means that it does change, right? So in other words, do I think that someone with Prader-Willi syndrome will have a different microbiome to someone without? Probably, because they're going to be eating different things. They're going to be under different hormonal control. There's going to be any number of different things that are there. But at the moment, all the research that I've looked at and read, and I'm not a microbiome expert, is that while it reflects health and reflects weight and reflects disease. It doesn't cause it's not causative, and therefore I don't think it's going to be used as a treatment for anything. There have been studies. This is a non-Prader-Willi obesity. There have been studies in which it's what we call, everyone, before you have your dinner, I'm sorry I'm talking about this now, what they call fecal microbiota transfer. I don't know if you know what that is. It's otherwise called a transfusion. I'm not making this up. It is really called a transfusion. It's when you actually take poo, yeah, I know, and actually transfer it into up the yin yang of something else to see if actually you can make a difference. Incidentally, if you purify the bugs and put it into a capsule, it's called a crapsule. I'm not making any of this stuff up either. Now, it's transfusions, fecal microbiota transfer, can be used to fix certain diseases, severe intestinal infections such as C. difficile and things like that. That does work. The studies that have been done in humans where they've taken the bug poos from lean people and stuff to put it into someone with obesity to see if anything happens, just no effect. There's no effect. So I do think that the as a cure, I think it's overstated, slightly hysterical, I think, but it is going to play an important role in reflection to health. Sorry, I didn't answer the question, but there we go. Transfusion. Everyone go Google it after.
Great, thank you. Thank you so much. I'm just wondering if there are any other, thank you for that question. We actually get asked quite often about microbiome and whether that has, whether there's a role to play. Just wondering, we're getting towards the end of our time. So is there any other questions that people would like to put forward? Now's the time. And Tania, do you have any other questions?
Oh gosh. No, I was just going to say that, you know, we've talked about people, not Prader-Willi specifically, but people in general getting bigger, and that our genes are basically the same. And so what's changed in the environment, and it's the food that we eat, and people talk about ultra processed foods. I don't particularly like that term because
neither do I.
Well, the way that, they say a yogurt is an ultra processed food, which is ridiculous. Yogurt's a great food. So there are foods that we can all think of that you see in the supermarket that grandma could never make, that are purely made in a factory. What are those roll-ups? That's one thing that, that you get in Australia. It's like a jelly, gum thing that's
fruit
a roll up, and
things
it's meant
that are roll
fruit
up.
roll. Yeah.
Okay.
I mean, I don't think it's ever seen any fruit. And then there's, well, when I had a kid, twisties. I love twisties. Goodness knows what's in twisties. But they've got the salt, they've got the sugar, they're soft, you can just eat lots and lots of it. It's got all that hedonic stuff that you're talking about. And we used to have it just as a little treat from time to time. Now it's packed in kids' lunch boxes. People, this discretionary sort of non-healthy, this food that has lots of calories with very little goodness in it, comprises a huge proportion of our diet. I can't remember the exact figure, but I think it's something like 30% of the calories that we ingest
50.
in Australia. 50, is it?
Yeah.
Okay. So it's frightening. So this food is not good for any of us, and it's certainly not good for our children, whether they have Prader-Willi or anything. And this is the big problem. You talked about it yesterday. Giles gave a very good talk at our institution yesterday. And if there's one thing that we need to change, it's the food that we eat. And interesting, a lot of. I mean, luckily, most people with Prader-Willi syndrome, I think, like fruit and vegetables. I've met some picky eaters. We're running out of time. I talk too much, but a lot of the patients I see in my obesity clinic don't actually like fruit and vegetables, or haven't eaten them, or don't know how to cook them. And it's interesting how poor the diets are. So those sorts of things are very important for all of us, including our relatives with Prader-Willi syndrome and children.
They really are. I've got one more question here. And I apologise. I've locked our dog in a room and he's barking his head off. And I don't know if you can hear him.
We can't hear him. No.
Fine. Okay. So there's a question, and look, this actually relates to something else I was thinking about. the International Prader-Willi Syndrome Organisation has done a lot of work on mental health, and Professor Tony Holland has recently said at our conference, it's very difficult to disentangle mental health and neurobehaviours from hyperphagia. I think, to me, this question sounds like it relates. So tell me what you think. What is the role of high cortisol levels on weight with relation to Prader-Willi syndrome?
I don't think it's going to be unique to Prader-Willi syndrome, because I think for a lot of people, high cortisol can drive a lot of food intake, but not everybody. Remember that's the first story I opened with about some people respond to stress by eating and some people respond to stress by not eating. I would imagine this is probably going to be the same in Prader-Willi syndrome, but I don't know. Okay, there's got to be some kind of phenotypic variability. But in non-Prader-Willi individuals, if you are responsive to cortisol for stress, then a number of different things happen. A, you respond to it by stress eating, comfort eating, to try and become unstressed. And so for a lot of people, food is a comfort. Okay, and that's the first thing. But we also know biologically that cortisol hits a population of neurons in our brain that also on top of that drives you to eat. Okay, on top of that, and also drives a lot of storage of fat into the central parts of the obesity, into the part of the bad fat, for lack of a better term. You got fat underneath the skin, and you got fat of the visceral fat around the organs. And so having chronically high levels of cortisol is not a great thing typically. Okay, typically. So the answer is, it's always best not to be stressed. It's easier said than done. I appreciate this. But yeah, that there is definite biological role for higher cortisol levels, because obviously cortisol is there biologically to prepare us for things. But if you chronically have a high cortisol, I think for a percentage of people, it will drive not only additional eating, but also probably drive storage of fuels into the wrong kind of fat.
That makes me just reflect on a conversation we had earlier today, Professor Yeo, where we talked about, that being hungry is unpleasant. It's painful. And so naturally then people with...
It's unpleasant whether or not you have Prader-Willi syndrome. Prader-Willi syndrome is more acute. Obviously, you respond to it more. But any one of us has been hungry. It's a deeply, really hungry, deeply unpleasant. And so you can see how it equates to mental health. People eat to stop being, for it to stop being unpleasant. And I can see how if you're unable to sate that unpleasantness, then you lose the enjoyment of food, because then you keep eating to try and kill the unpleasantness, but you don't get the joy from it. And that's a real thing. It's a real thing.
Gosh. And I feel like we've now come back to where Tania was talking earlier today about some people with Prader-Willi seem to eat not because they're seeking the pleasure, but just to feel that. We have one last question then related, just leading on from that. So how can we reduce stress for our people with Prader-Willi syndrome? This person says, I know it's a difficult question, but is there something we can do?
So I'll let Tania take the last thought on this. I'm just going to give you, because she sees Prader-Willi patients, I'm assuming she knows, but I can give you an answer of what I think for a lot of people, whether or not you have Prader-Willi syndrome or not, unpredictability is stressful. This is what happened with COVID, right? The problem with COVID is we were all stuck at home. We didn't know when the hell it was going to go. We didn't know what was happening. Was the world going to end? And when was it going to end? And so the initial stages, you had to do weird things like line up to go to the supermarket. It's all very stressful. But I do think it's true that for many people, and many people with Prader-Willi syndrome, that if you don't have some kind of prediction or predictive schedule, that is then very stressful. So I just get the feeling you want to make sure that the habits that are there clearly are better and are healthier, but are predictable. Certainly I am. My wife thinks I'm a creature of habit. I am a creature of habit because I get stressed when things are a little bit when they're not predictable. I like things just so, and I can imagine that's going to be the case for a lot of people, particularly if you have Prader-Willi syndrome.
Well, for somebody who doesn't look after people with Prader-Willi syndrome, you have made that's spot on.
Oh, good.
You get full marks. I couldn't have answered the question better. Yeah. So having a routine, having You know when your meals are, you know what you're going to get, you know what's happening on Monday, Tuesday, Wednesday. You know, changing a medication, which sometimes needs to happen, that's just one example, is very stressful for a lot of the patients. And so I don't always do it immediately. I'll explain why, and I'll say, well, we won't change it this time. You think about it, and we'll discuss it next time, and then we'll see. Because often those changes aren't, they're not going to make a huge difference. So, I mean, obviously, if something has to happen immediately, it does. But change is not something that people with Prader-Willi like, and it does stress them a lot. And if we can keep the environment or keep their routines as regular as possible, that does seem to make a big difference. Also important is knowing how to manage a situation when behaviour does get out of hand. And I see parents or carers with their individual managing it so well, and others doing the completely wrong things that will rev the patients up, and that can make an enormous difference. How you assist somebody who is getting upset when they are getting upset. Very hard.
Yes. Well, I have found this a really incredibly informative and very engaging conversation. I want to thank you both, Professor Yeo and Associate Professor Markovic. Thank you both for your time. It's only because of the goodness and the generosity of people like yourselves that we can continue to offer this type of incredible information to our community. And we know that they really appreciate it and they really value it. So thank you so much.
You're very welcome. Thanks for
Pleasure.
having me, and thanks everyone for listening.
Yeah, and thanks to the audience and for the great questions. We really appreciate it.
Thank you.
Thanks, everyone, and good night.
Thanks for giving me the chance to hear Giles again. Thanks.
Lovely to meet you, Tania. And lovely to meet you, Kathleen.
Lovely to meet you too. It's been a real pleasure.
Yeah,
Thank you.
Peace.
See you guys
Let's
later.
hope.
Bye.
Bye.
Bye.